Let's continue with our nervous system, our neurohistology, and we're on the axon. And two structures that are within the axon, neurotubules and neurofilaments are important because they give structure to the axon. If a process has to be so long, these provide structure and can help direct what we call axoplasmic flow. The cytoplasm within the axon is flowing from soma to synapse. So these provide, well, we won't provide, they're, use that word before. They direct axoplasmic flow. Now, let's continue with our myelin that we had introduced. Myelin is formed from two kinds of cells. In the PNS, myelin formed by what? Schwann cells, right. Schwann cells, after Mr. Schwann. In the CNS, myelin is formed from what? Oligodendrocytes, right. I'm not going to write formed again, but oligodendrocytes. So a Schwann cell, as it, let's put our typical neuron in here. And this is our axon. We'll see that the Schwann cell, many Schwann cells can myelinate a single axon. And we'll put a few in here. They wind around. So this will represent the nucleus of a Schwann cell. Here's another one. Just trying to show that the myelin is laid down in layers. Another nucleus. So these are examples of Schwann cells that have wrapped around and formed so many Schwann cells per axon. Now the distance between the Schwann cells is a very important area. On the nerve fiber here, this distance which has no myelin, this is referred to as the node of Ranvier, node of Ranvier. And there's no myelin. It's just the gap between myelin cells. And it's important with the conduction of the impulse because the impulse will go and between nodes of Ranvier. So the impulse they call it jumps between nodes of Ranvier. So we've already said the more myelin, the more rapid the impulse. Now with the oligo, one oligo can myelinate many fibers. Quite a different picture. So the other formation of myelin comes from oligodendrocytes. And one oligo, as we call them, working in the field, can myelinate many axons. So when we get these demyelinating diseases, you want to be aware that there are two kinds of cells, two kinds of processes by which they myelinate. Now, does anybody know anybody with a demyelinating disease? What's the worst nerve you want to get demyelinating? What's one I just wrote on the board, on the other board? Phrenic nerve. Why is it the worst? There goes your diaphragm, there goes your breathing, right. So you can get demyelination throughout many areas, but catch it in the phrenic, and that's drastic. So let's say a word about dendrites then. And we said you could have one dendrite, or you could have thousands. Just think of the receptive surface when you have thousands, the integration that has to go on within a single cell. Now, dendrites can increase their surface area by having what are called spines. Have you heard of dendritic spines? Dendritic spines increase, I'll show you pictures of them, the surface area of the dendrite. They're quite fascinating little structures. If I have a dendrite, then I'll have a little spine come out. This will represent a dendritic spine. As innocuous as it seems, it can receive as many as 9 synapses on it. An area of very rich integration on a dendritic spine. I'll show you an EM electron micrograph with 9 synapses. Can have, we'll put one or many synapses on a spine. So they're not just innocent little bystanders. They are very active. In fact, they can change their structure in 30 seconds. And I called Floyd Bloom, who was head of science before, just a week ago, just to ask him, do we know anything more about when they disappear, if they leave a molecular configuration there, or whether they lose it and they have to start from scratch. But spines are, as some people say, very important, and they are different shapes and sizes, but they're a dynamic part that most people aren't aware of in your lower biological courses, but when you go along, you'll get acquainted with them. Now, we want to look at glial cells. Can I take this off? glial cells. Briefly, we can say that they're the metabolic and structural support cell of the nerve cell. Metabolic, somebody just asks, what are they? Metabolic and structural support cell for the neuron. They need each other. There are what we call macroglia, large macroglia, and microglia. Types of macroglia will be astrocytes. They look like stars. Astrocytes. Astronomy. And other kind of macroglia will be the oligodendroglia. Now the macroglia are derived from ectoderm. For those who haven't had any embryology, there are 3 germ layers from which all tissues of the body are derived. And it's interesting when you're trying to find out functions of cells, do they all have the same derivative or are they different? So this is just in embryo. All tissues from either ectoderm, mesoderm, or endoderm. Nerve cells come from ectoderm, and so do macroglia. Microglia come from mesoderm. If microglia come from mesoderm, how do they get into the central nervous system, which is an ectodermal derivative? The microglia will migrate from the bone marrow. Now you know what bone marrow is. Along blood vessels to the CNS. So what we'd like to do then is look at astrocytes. Why do you need astrocytes? What are they doing for you? How are you going to tell an astrocyte from an oligo? So astrocytes. So as we said, star-shaped. So they'll have all processes. They're different kinds, so it's sort of difficult to do a single kind. But they've got all these processes. This is cell body. Astrocytes, many processes. And all are alike, different from a nerve cell where you've got dendrites and axons. Many processes and all alike. Now, why do you have astrocytes? What are their functions? First very important function They initiate the formation of the blood-brain barrier. Initiate the formation. They do not make up the blood-brain barrier, they initiate the formation of it. Very important distinction. So let's put it here so we have more room. Initiate the formation of the blood-brain barrier. What in the world is that? Again, we abbreviate it. Will be tight junctions between the endothelial cells of the capillaries in the brain. Not everything that the rest of the tissues get does the brain want. It wants to limit and be able to choose what it wants to come in. And so we have a barrier, and the barrier equals the tight junction between endothelial cells in brain capillaries. Other capillaries don't have tight junctions. What else? The astrocytes help form the peel glial membrane. Peel glial membrane. They help form peel glial membrane. Now, what in the world is that? We talked about the membranes that protect the central nervous system. Well, the pia is the one that's adherent to the CNS. So briefly, if we do this, and just call this brain, we'll have a peel membrane just adherent to the brain tissue. So this is pia. What does pia mean? What's its full name? Pia mater versus dura mater. Pia is gentle, gentle mother. And now then we'll have, let's just blow up an astrocyte inside the brain. And it'll take its process over and put it adjacent to the pia, called a foot process. Another astrocyte, come over, put a foot process. So the peel glial membrane, astrocytes in pink, is the foot process of astrocyte and the peel membrane. And this membrane surrounds the whole CNS, spinal cord and brain. So it's for protection. Get an ice pick that stabs through your brain, immediately these glial cells will multiply and form scar tissue. So it's for protection. Very important. Though it causes problems when you want regeneration because you've got scar forming, it's very important to protect. Now, why else do we have astrocytes? Every time your nerves fire, they liberate potassium. They pick up that excess potassium. So after neuronal firing. The astrocyte picks up excess potassium. I'll write it out so it's not confusing. Loads and loads of functions here. Do you ever wonder, those who've studied the nervous system have seen that when a synapse fires and the transmitter goes across, why doesn't it go sideways? There's nothing on the sides of the synapse. You have astrocytic feet. They prevent the transmitter from going sideways. So we could go on and on and on with the functions here. Very dynamic. Have you ever known you had astrocytes? Ever thought about them? No? They're very precious. Astrocytoma, has anybody who's had a tumor of astrocytes? Yes, the glioblastoma multiforme? Good, because once it's diagnosed, you have no more than a year to live. It's really a deadly. Brain tumors are primarily gliomas. They're not neuromas. So glial cells, don't shun them. They are terribly important. Glioma, that's tumor of glia. No, I get off on tangents, but my brother had a glioma, had an astrocytoma, and the first thing the doctor said, did he smoke? And I said, yes. It just sort of fit, just like we learned with cancer of the lung, right? Gliomas, too, with smoking. You never hear about them because people don't know glial cells. So, anyhow, you have lots of things to say. Oh, one of the most important here, I better put it because it is important. They serve as a scaffolding for neural migration during development. Very important. If cells don't migrate, aren't you lucky? All your cortices, all your cells have migrated into position. Major function is to get them in the right place. You get a flu virus at a certain time during gestation, you can knock them off course. It's amazing. Such a dynamic system. All right, now we'll go to oligos. We've already said what oligos do primarily. We don't know much about oligos' functions other than forming myelin. But I will mention one thing. Oligo functions, repeat, form myelin. But also they serve as satellite cells to blood vessels and neurons. Serve as satellite cells to blood vessels and neurons. And we're not quite sure what they're doing there. But you see them there all the time, what they're exchanging with those blood vessels, what kind of nutrients, what kind of ions, whatever. But that's a major cell, and it has that position. Myelin, sure, we know. Then I mentioned the microglia. I'll say one more word about them. We said they were mesodermal. But we didn't say anything about their function. They're very small until activated, until there's an injury. Until activated by injury. They're just sitting there sort of waiting for an accident to happen. But when it does, then they become phagocytes, become phagocytic. What popular disease with aging are nerve cells degenerating today? Alzheimer's. Many microglia are present in Alzheimer brains. Then there are lots of other glial cells. Little by little, we'll encounter them. But these are the major. Do you like glial cells? Do you? I don't think this little fellow that I'm talking to right here does. Do you? You're not interested in glial cells. Do you like neurons? Yeah, okay. Well, it's my job then as a teacher so that you become acquainted with both, because one without the other is not very useful. And that's a problem with many of our theoretical people. They go only to neurons, and they don't realize they should know the glial cells as well as the neurons. Respect to them. I mean, I'm not condemning them, but it's so important. So now, we want to look at the synapse. And many of you probably had your synapses in basic courses, but for those who haven't, because there are quite a few who haven't, this is the structural and functional unit of the nerve cell. This is where the dynamics takes place, where the variation is. All kinds of neurotransmitters. So, synapse. Structural and functional unit of the nervous system. So, we're going to follow an impulse down an axon to a dendrite. Let's put them in here sort of in large form. This is the end of an axon, the beginning of a dendrite. Axon, dendrite, and this junction between the two is our synapse. And we'll first follow down an electrical impulse, because sometimes somebody wants to know how a nerve works, you can use your arms and your hands very readily. This would be the main axon coming down. It's all electrical down here. As it gets to the terminals, then it's chemicals. Chemicals cross the gap, stimulate, and you have the electrical impulse going on. So we're going to see then how the electrical impulse is coming down. So one is the electrical impulse coming down the fiber, down axon. And then we come to what's called the presynaptic terminal. If this is my synapse in here, this is my presynaptic terminal. And the presynaptic terminal is filled with little packets of neurotransmitter, vesicles. So we're going to put in vesicles. So 3 will be vesicles of neurotransmitters. Neurotransmitters. T-R-A-N-S-M-I-T-T-E-R-S. T-R-A-N-S-M-I-T-T-E-R-S. The neurotransmitters are formed in the presynaptic terminal. Formed in presynaptic terminal. They also contain neuromodulators. For some transmitters to work, they have to be in a certain environment. So you have the chemicals that modulate the environment so the neurotransmitters can work. So we also have vesicles of neurotransmitters and neuromodulators. When we first started this work, there were maybe 5 known neurotransmitters. Now there are 100 if you combine all the neuromodulators with the neurotransmitters. The synapse is so complex now. It used to be so simple, like everything else in life. All right, so you have the neuromodulators are formed in the soma of the nerve cell. How do they get down to the presynaptic terminal? What conveys them down? The exoplasmic flow. You've got to have some transportation from the soma to the terminal. So that's why you have this flowing exoplasm. Once you've seen people collect exoplasm out of the giant squid, you never forget it, because there's so much exoplasm in that big neuron. It just drips off. So you know that it's a dynamic structure. So now what's going to happen? We've got our vesicles. They're sitting there. We've got to activate them, get them close to the terminal here. So we have, when the electric current's coming down, we have calcium ion out here. So 4 will be coming in here, and 4, can I take off this or should I go to the other board? You're finished with this side? Are you? Great. Keep it close if I can. I mean, just think, for every movement, every eye blink, all of this is taking place. There's some now, right? All right, so 4 represents a calcium ion. When the electrical impulse comes down, the calcium ion comes into the terminal and sets up a substructure. To guide the vesicles down to the membrane. So calcium enters presynaptic terminal. Sets up substructure. To guide vesicles to presynaptic membrane. And once they get there, then they fuse with the membrane and discharge their contents got some contents here. Just make it neurotransmitter. And calcium enters, sets up the substructure to guide the vesicles to the presynaptic membrane, to release the neurotransmitter. They open up and put the neurotransmitter in the synaptic cleft. This is my synaptic cleft. And what are they going to do? Well, they've got to react with receptors on the postsynaptic membrane. Receptors on postsynaptic membrane. And will open ion channels, two open ion channels, and that's their function. That's what the neurotransmitters do. Open those channels. And when the channels open then ions rush in. Let's just take calcium ion can go in to my postsynaptic ending. Or terminal. To act on, what are they going to act on? Enzymes. Or genes. Say they'll act on genes for protein formation. We can just come out for those. For protein formation. And what are we going to do with that protein? We can build a bigger terminal to enlarge the terminal. We find with our enriched environments, when our rats go to school, they get bigger postsynaptic terminals. When they don't, they shrink. You see why I work so hard for years to get big? Because the bigger those postsynaptic terminals, the better the learners those rats are. If they can do it, you can do it. Right? So for protein formation, for neuronal structure, as we'll just put in general. Neuronal structure. So that gives you an idea of how a synapse works. But now you say, what's going to happen to this transmitter that's up here in the cleft? We don't want it to keep firing. So it can break down. The transmitter in the cleft can break down, or it can recycle and go back into the presynaptic terminal. So neurotransmitter in cleft can break down. Technical term is to hydrolyze. Hydrolyze. Or can recycle. We're all aware of how important recycling is in today's world. So how's it going to recycle? How's it going to get back into the presynaptic terminal? For recycling, the neurotransmitter will hop a carrier. There's some carriers in that synaptic cleft. Neurotransmitter? What does that mean? Are you okay? Well, communicate with me. If you're not okay, tell me where to stop. All right? It's all basic. It's happening in your system every second of your existence. The neurotransmitter hops on a carrier. And the carrier takes it back to the presynaptic terminal. Back to presynaptic terminal. That's presynaptic. Let's write it better. Terminal. Now, why do we stress this? Thank you. Good, I can make my point. How many of you have taken cocaine? Just want to see if you're awake. If there's an empty carrier in there, and you're taking cocaine, cocaine will hop on that carrier and block the reuptake of the transmitter, dopamine. It's dopamine that has these carriers. So you don't want to take cocaine. Oh boy, sorry. Sometimes the erasers work and sometimes they don't. So dopamine hops carrier if available. Cocaine hops carrier if available. So what happens if the cocaine hops a carrier, then the dopamine has none, so the dopamine stays in the cleft and can keep firing. Give you a high. Soon wears out. Needs more. All right, let's look at some slides. This is a picture we showed last time, but you hadn't had the myelin. So let's say, if you're shown this slide, are we in the CNS or the PNS? PNS, good for you, because you've got your single Schwann cells here. You've got the node of Ranvier. When you're studying electrophysiology, you'll see the response at the node of Ranvier. We said saltatory conduction goes between nodes of Ranvier. But these are Schwann cells that have wrapped around forming myelin. Myelin consists of a lipoprotein within the Schwann cell. It's in segments. Next one. This is just to show dendritic trees, the variation. We showed it last time. But you can appreciate now how many synapses can occur for that single cerebellar cell to fire. Isn't that amazing? In the next one. Now these are dendritic spines. You may do a preparation with this method and you'll say, well, look at all the precipitant I have on my dendrites. That's not precipitant. Those are direct little spines, each one receiving synapses to the main fiber. Terribly important. In the next one, I'll say when Francis Crick first thought he's coming into the nervous system to get his second Nobel Prize, he was going to work on spines because he thought that's where memory formation was. But if they're changing in 30 seconds, See, you need to know your neuroanatomy, right? Here's a spine from the electron micrograph. Here's a synaptic terminal with all the vesicles, all the mitochondria. Here's a synaptic terminal, all the vesicles. There's presynaptic terminal, postsynaptic terminal. Here's another one here. These are all mitochondria, but you can count 9 synapses on a single spine. Next one. Next one, please. I think we had that one. These are astrocytes. These are what we call protoplasmic astrocytes. They're all kinds. It's got this film around it. That's natural. It's got many processes. And somebody showed on the front cover of Scientific American once that all of these form a syncytium. They're all connected. I've wondered with astronomers, are all stars connected somehow? But the astrocytes of the brain supposedly are. Next one. And these are oligos. Oligo means few, so few branches. Here's an oligo. Quite different from the astrocytes. In the next one. And this just shows many of the things that an astrocyte can do. Here's an astrocyte. Here's a Schwann cell. Schwann cell on a fiber. Here's a Schwann cell. Here's a node of Ranvier. To prevent things from leaking out here, there's an astrocytic process. Here's a synapse. With my synaptic junction, here's an astrocytic process. These are the ventricles, the channels. These are ependymoglia. Here's an astrocytic process. We should have pial glial out here. Here's an astrocytic process. Here's an astrocyte around the blood vessel, on the soma. They're everywhere. You don't want an astrocytic tumor. In the next one, here's an artist's rendition. Here's a spine. Here's a presynaptic terminal, losing the myelin. Cut it open. Here's our synapse, presynaptic terminal, postsynaptic terminal. Here's our myelin. Myelin stops before it gets down to the presynaptic terminal and expands full of vesicles. In the next one. Next one, please. And this just shows how this is how synapses grow stronger with use. You have the electrical impulse coming down. Here's your presynaptic terminal. Here are your vesicles filled with transmitter. The calcium will run in. These are glutamate, so that you've heard of glutamate. It's the most common neurotransmitter in your brain. So it's here. You get calcium coming in. These will march right down to the presynaptic membrane, pour out their contents, open a channel. Calcium rushes in, can activate enzymes, alter protein composition of the synapse, switch on genes, and of course, continue with the impulse down the fiber. But releasing retrograde messenger, probably nitric oxide, man at UCLA got the Nobel Prize for that one. He drives his yellow Porsche and his license plate says Nobel. Just to let you know, I want you to smile a little bit because sometimes this gets heavy, and that way you can relax, that there's always a little story behind everything. Next one. And this shows, what's this? Ooh, here's an Nissl everywhere but here. Axon hillock, that's an axon coming off a nerve cell. Next one. These are an artist's rendition. Here, all the Nissl substance everywhere, dendrites, but not in an axon. These are all glia surrounding nerve cells. Next one. Next one, please. And this just shows you, when you were born, look at, you have few dendrites. But look at a 2-year-old. Not amazing? No wonder they're so difficult to deal with. But it just lets you know how important dendrites are. We'll stop there.